Welcome students. Today we are going to discuss genetic code. F.S.C. Crick postulated the existence of genetic code, the set of all codons that specify the 20 amino acids, the number and sequence of bases in mRNA specifying an amino acid is known as codon. The codons are usually presented in a language of adenine, guanine, cytosine, uracil. If a single nucleotide is capable of coding for amino acid, that is singlet code, only 4 codons are established and doublet code would be 16 codons, which are not enough to code for 20 amino acids. However, if 3 nucleotides code for 1 amino acid, triplet code, as many as 64 codons become available for 20 amino acids. The 64 triplets would be enough to code for 20 amino acids. Crick 1961 provided the first experimental evidence in support to the concept of triplet code of mRNA. A chemical called proflavin was given to T4 bacteriophage, which could either add or delete a base in its DNA molecule, thus damaging the virus and resulting in an altered or mutant form of the virus. When insertion or deletion of a single or double base pairs occurs, the bacteriophage ceased the normal function. In addition, when 3 base pairs were added or deleted in the T4 DNA, the bacteriophage performed the normal function. Based on this experiment, they concluded that genetic code is a triplet code because due to addition or deletion of single or double base pairs, the reading sequence was changed, whereas it was returned to normal with the addition or deletion of third nucleotide. Accordingly, a codon dictionary has been prepared and a relationship of some 61 codons has been established to certain specific amino acids. The remaining 3 codons, UAA also called ochre, UAG also called amber, and UGA also called opal, do not code for specific amino acid. And before the function of these codons was discovered, they were called nonsense codons. The 3 codons UAA, UAG and UGA, whenever present in mRNA, would bring about termination of the polypeptide chain and are given the name stop or termination codon. Since there are more codons than the amino acids, and as a result, almost all amino acids are represented by more than one codon. The only exception are methionine and tryptophan. The codons that have same meanings are called synonyms. Thus, multiple codons must code the same amino acid. This is called degeneracy in the genetic code. Now, we will discuss features of the genetic code. Number 1, triplet code. A singlet code means one-to-one correspondence between nucleotides and amino acids and has been ruled out by the biologists as the nucleic acid code only for 4 types of amino acids. In doublet code, 2 nucleotides code for 1 amino acid and only 16 amino acids will be coded and is insufficient to code for 20 amino acids. In triplet code, 3 nucleotides code for 1 amino acid is thus the smallest coding unit that could accommodate 20 amino acids. So, the triplet code fulfills the requirement of coding all the 20 amino acids. Number 2, non-ambiguity of the code. It means that there is no ambiguity about a particular codon. A codon will always code for a particular amino acid. However, the amino acid can be coded by more than one codon, but same codon shall never code for two different amino acids. There is some ambiguity when AUG and GUG are taken into consideration. Both may code for methionine as initiating codon, although GUG is meant for valine. Number 3, genetic code is universal. The genetic code is the same in almost all organisms. For example, the codon AGA specifies the amino acid arginine in bacteria, humans, and all other organisms whose genetic code has been studied. The universality of the genetic code is among the strongest evidence that all living things share a common evolutionary heritage. The universality of the code argues that it must have been established very early in evolution. Perhaps the code started in a primitive form in which a small number of codons were used to represent comparatively few amino acids, possibly even with one codon corresponding to any member of group of amino acids. More precise, codon means an additional amino acid could have been introduced later. Evolution of the code could have become frozen at a point at which the system had become so complex that even changes in the codon meaning would disrupt existing proteins by substituting unacceptable amino acids. Its universality implies that this must have happened at such an early stage that all living organisms are descended from a single pool of primitive cells in which this occurred. Because the code is universal, genes transcribed from one organism can be translated in another. Similarly, genes can be transferred from one organism to another and be successfully transcribed and translated in their new host. This universal gene expression is central to many of the advances of genetic engineering. Many commercial products, such as the insulin used to treat diabetes, are now manufactured by placing human genes into bacteria, which serve as tiny factories to turn out prodigious quantities of insulin. Number 4, genetic code is degenerate. All the amino acids except methionine and tryptophan are specified by more than one codon. A non-degenerate code is one where one codon codes for one amino acid. That is, 20 codons code for 20 amino acids and rest 44 codons are useless. But it is not the case. This occurrence of more than one codon per amino acid is called degeneracy. The degeneracy in the genetic code is not at random. Instead, it is highly ordered. Usually, the multiple codons specifying an amino acid differ by only one base, the third or 3' base of the codon. Because of the degeneracy of the genetic code, there must be either several different tRNA that recognize the different codons specifying a given amino acid, or the anticodon of a given tRNA must be able to base pair with several different codons. The degeneracy is primarily of two types. Number 1, partial degeneracy, and number 2, complete degeneracy. Now, what is partial degeneracy and complete degeneracy? Partial degeneracy occurs when the first two nucleotides are identical, but the third or 3' base of the codon differs. The third base may be one of the two pyrimidines, uracil or cytosine, and the codon will still specify the same amino acid. For example, CUU, CUC, code for leucine. Similarly, purines, adenine and guanine are often interchangeable for the third base of a codon. For example, GUA, GUG, code for valine. In case of complete degeneracy, any of the four bases may be present at the third position in the codon, and the codon will still specify the same amino acid. For example, UCU, UCC, UCA and UCG, code for serine. Now, what is importance of degeneracy in genetic code? Degeneracy in the genetic code evolved as way of minimizing mutational lethality. If the degeneracy is of the type that leads to the replacement by equivalent amino acids, the small accidental mutational changes are much less damaging than that would occur under a non-degenerate code. Thus, degeneracy contributes favorably to genetic stability. The vowel or third base of the codon contributes to specificity because it pairs loosely with its corresponding base in the anticodon. It permits rapid dissociation of the tRNA from its codon during protein synthesis. If all three bases of codons engaged in strong Watson-Crick pairing with the three bases of the anticodon, tRNAs would dissociate too slowly, and this would severely limit the rate of protein synthesis. Thus, codon-anticodon interactions balance the requirements for accuracy and speed. Now, what is Wobble hypothesis? Transfer RNA base pair with mRNA codons by means of a 3-base sequence on the tRNA called the anticodon. The first base of the codon in mRNA 5' to 3' direction pairs with the third base of the anticodon. If the anticodon triplet of tRNA recognized only one codon through Watson-Crick base pairing, cells would have a different tRNA for each codon of an amino acid. This is not the case. For example, the anticodons in some tRNA contain the nucleotide inosinate designated as I, which contains the uncommon base hypoxanthine. Inosinate can form hydrogen bonds with three different nucleotides: uracil, cytosine and adenine. Although these base pairing are much weaker than the hydrogen bonding between the Watson-Crick base pairs guanine cytosine and adenine uracil. Examination of these and other codon-anticodon pairing led Crick to conclude that the third base of most codons pairs rather loosely with the corresponding base of its anticodon. The third base of such codon and the first base of their corresponding anticodon vowel. Crick in 1965 proposed a hypothesis known as Wobble hypothesis to explain this phenomenon. The first two bases of mRNA codon always form strong Watson-Crick base pairing with the corresponding bases of the tRNA anticodon and confer most of the coding specificity. The first base of the anticodon determines the number of codons recognized by the tRNA. when the first base of the anticodon is cytosine or adenine. Base pairing is specific and only one codon is recognized by that tRNA. When the first base is uracil or guanine, binding is less specific and two different codons may be read. When inosine is the first nucleotide of an anticodon, three different codons can be recognized. When an amino acid is specified by several different codons, the codons that differ in either of the first two bases require different tRNAs. A minimum of 32 tRNAs are required to translate all 61 codons. Number 5. The genetic code has polarity. The code has polarity, that is, it is read between the fixed start and stop codons. The start codon is also known as initiation codon and stop codon as chain termination codon. The message of mRNA is read in 5' to 3' direction. The polypeptide chain is synthesized from amino end to carboxyl end. Now, what is chain initiation codon and chain termination codon? The codon present in the beginning of the cistron is known as initiation codon. It marks the beginning of message for polypeptide chain. The initiation codon is A, U, G in majority of cases. It codes for amino acid methionine. Rarely, G, U, G also acts as initiation codon in bacterial protein synthesis. G, U, G code for valine. 3 of the 64 codons do not code for specific amino acids. These codons are UAG, UAA and UGA. These bring about termination of polypeptide and are therefore called as termination codons. Number 6. Genetic code is non-overlapping and commaless. The genetic code is non-overlapping means that a base in mRNA is not used for two different codons. Commaless code means that no punctuations are between the codons. In other words, the code is read from fixed starting point as a continuous sequence of bases taken 3 at a time. For example, A, B, C, D, E, F, G, H, I, J, K, L is read as A, B, C, D, E, F, G, H, I and so on without any punctuation between the codons. When one amino acid is coded, the second amino acid will be automatically coded by the next 3 letters and no letter are wasted for telling that one amino acid has been coded and that now second should be coded. If 1 or 2 nucleotides are either deleted from or added to the interior of a message sequence, a frame shift mutation occurs and the reading frame is altered. The resulting amino acid sequence may become radically different from this point onwards. Collinearity of genetic code. Genetic code represents sequences of codons in mRNA and corresponding amino acid residues of a polypeptide chain are arranged in the same linear sequence, that is, the code is collinear with amino acid sequence in a polypeptide chain. The translation of mRNA occurs concomitantly with its transcription. mRNA is translated in the same direction as that in which it is synthesized. RNA chain grows from 5' to 3' end and the translation of mRNA too goes from 5' to 3' end. The protein chains grow from their amino terminal end. It has been shown that the amino acid closer to the amino terminus is represented by a codon closer to the 5' end of the corresponding mRNA. Now, cracking of genetic code or cryptanalysis of genetic code or deciphering the genetic code. The deciphering of the genetic code means which codons specify which amino acid, how the code is punctuated, whether different species use the same or different codons. In 1950s, it was theoretically accepted that genetic code should be triplet in nature, and it was not possible to say which out of 64 codons code for which amino acid. The first major breakthrough came in 1961 when M. W. Nirenberg and J. H. Mathey synthesized RNA using only uracil, so that all along the length of mRNA there was no other base and the only possible triplet was U, U, U. When such a poly-U RNA was used in cell-free synthesis of a polypeptide, only polyphenylalanine was synthesized, meaning that only amino acid coded was phenylalanine. It was therefore immediately concluded that the triplet U, U, U coded the amino acid phenylalanine. Subsequently, poly A gave polylysine and poly C gave polyproline. Therefore, AAA was assigned to lysine and CCC was assigned to proline. In 1964, M. W. Nirenberg and P. Lieder, through their binding technique, found that if a synthetic trinucleotide for a known sequence is used with ribosome and a particular amino acyl tRNA, these will form a complex provided the used codes for the amino acid attached to the given amino acyl tRNA. In a process, for example, codon 1 plus ribosome plus amino acid tRNA forms a complex ribosome codon 1 amino acid 1 tRNA 1 complex. If given amino acid 1 is used with a given codon 1 and the formation of the complex is detected, this would prove that the given codon codes for the given amino acid. It was also observed that while the free amino acid tRNA passes through nitrocellulose membrane, the ribosome codon amino acid tRNA complex adsorbs on the such a membrane. If in a particular mixture only one of the amino acid is made radioactive, then the presence or absence of the radioactivity on the nitrocellulose membrane will show whether there is a relationship between the codon and the amino acid which was made radioactive. For example, 20 samples of a mixture of all 20 amino acids may be taken, and in each sample one amino acid is made radioactive in such a manner that each and every amino acid is made radioactive in one sample or the other, and no two samples have same radioactive amino acid. A particular sample would be then known by its radioactive amino acid. Now, tRNAs and ribosomes are mixed with each sample, and the same codon is used for complex formation in all 20 cases. When the mixture is poured on the nitrocellulose membrane, radioactivity on the nitrocellulose membrane will be observed only when the radioactive amino acid is taking part in the formation of the complex. Since in each sample the radioactive amino acid is known, it would be possible to detect the amino acid coded by a single codon by the presence of radioactivity on the membrane. Such a treatment was given by Nirenberg and his co-workers to all the 64 synthetic codons, and their respective amino acids were identified. By this technique, Nirenberg and his co-workers cracked 45 codons for amino acid arginine, alanine, methionine, proline, tryptophan, tyrosine, serine, and valine. H.G. Khorana, 1961, at the same time devised an ingenious technique for cracking the genetic code. Khorana and his co-workers could prepare polyribonucleotides with known repeating sequences. A repeating sequence means that if Cu are two bases, these will be repeatedly present throughout the length as follows: Cu, 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 and so on. In a similar manner, if ACU are three bases, they will be present repeatedly as follows: ACU, 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 and so on. Such copolymers will direct the incorporation of amino acids in a manner which can be theoretically predicted. For instance, in Cu, CUC, UCU, CUC, UCU, CUC, UCU, only two codons are possible, and these are CUC and UCU. Moreover, these codons are present in alternating sequence. The result would be that the polypeptide formed would have only two amino acids in alternating sequences. We may similarly consider a repeating sequence of three bases, for example ACG. Depending upon where the reading is started, three kinds of homopolypeptides are expected. Actual codon assignment, that is, to find out which of three codons codes for which amino acid, would depend upon the previous information available regarding the composition of bases in different codons. On the basis of the above techniques, a complete genetic code dictionary could be prepared. Now, we will discuss mitochondrial genetic code. In 1979, scientists began to determine the complete nucleotide sequences of the mitochondrial genomes in humans and mice. It came as a shock when these scientists learned that the genetic code used by these mammalian mitochondria was not quite the same as the universal code that has become so familiar to biologists. In the mitochondrial genomes, what should have been a stop codon UGA read the amino acid tryptophan. AUA was read as methionine rather than isoleucine, and AGA and AGG were read as stop rather than arginine. Furthermore, minor differences from the universal code have also been found in genomes of chloroplasts and ciliates. Thus, it appears that the genetic code is not quite universal. Some time ago, presumably after they began their endosymbiotic existence, mitochondria and chloroplasts began to read the code differently, particularly the portion of the code associated with stop signals. The major difference between the universal codon and mammalian mitochondrial code are: number one termination codon UGA and two arginine codons AGA and AGG in universal code. In mammalian mitochondrial code, UGA codes for tryptophan and AGA and AGG codes for stop signals. Number two, number of tRNAs is 22 in mammalian mitochondria code, while it is 55 in E. coli. Number 3. In Universal Genetic Code CUN, N stands for nucleotide, codes for leucine, and in mitochondrial code CUN codes for threonine in yeast. Number 4. UAG anticodon is amino acylated by leucine in Universal Genetic Code and UAG anticodon in yeast mitochondria accepts threonine. Different genetic codes have been identified in protozoa, Mycoplasma capricolum, in 1986. In this, UAA and UAG code for glutamine instead of stop signals. Dear students, this is all about genetic code. Thank you very much.